Biopython library. But before we go into that, any questions about what we discussed day before yesterday? Did you practice that? Any doubts? Any questions? Anyone? Nothing? Okay, good. So let's move on to the Biopython part. Let me just unshare the slides. Just a second. Okay. So as we discussed that when we are working, I hope you have already installed Python, because it would be better if you are working on a local installation than on Google Colab.
Yes.
Okay. So is there a question? No. Okay. So as we discussed that in Python there are many libraries, and these libraries helps us to add functionality to the core Python. So let me just give you an introduction on how exactly this library concept works, and if you want to use any library, what's the basic syntax. So let's say I do print sqrt4. Now sqrt4 is a function to calculate square root. So if I run this, I get an error. An error says that this name sqrt is not defined. Now why this error appears is because in the core Python there is no such function as sqrt. So if you
Excuse me,
try
sir.
to, yes,
I'm sorry. Excuse me.
I just what?
So I just joined. I just want to confirm one thing. Does the class start from 6:50 or 7?
7.
Okay.
Okay. All right.
Okay. Please continue.
Okay. So see these. Okay. So these functions, this sqrt function is not there in the core Python. So, but it's there in NumPy library. I know that. So to use this, I'll first do import NumPy. So now when I do import NumPy, I have all the functions that are available in the NumPy library available to me. And then here I need to do numpy.sqrt. So this syntax means that there is an sqrt function in the NumPy library and we want to use that function. So now if I run this, it gives us the square root. Okay, so this is the basic concept of importing a library and using functions in that library. Now let's go to Biopython. First of all, if you have installed Biopython, then it's good. Otherwise, you can just use this command pip install Biopython, and you can just paste it in this cell, empty cell, and you can install right here itself.
Sir, can you explain it again, this one?
This
Why
NumPy.
we imported? Yes, sir.
Okay, so see, whenever we want to use some library, the first thing we need to do is we need to import that library into our program. Just installing that library is not enough. So let's say if you have installed NumPy, so it won't be available to you by default. So you need to import it. Now the reason why you import it is because let's say if you do print, okay, now this would work because I have already imported the library. So so I do import name of the library, whatever library I want to import, and then I have access to all the function within that library. Now this sqrt is a function in NumPy library. It's not there in core Python. So once I have imported NumPy library, then I can say print numpy.sqrt and then the number for which we want to calculate square root. So now it would give me the square root. If I don't import NumPy and straight away try to run sqrt parenthesis 4, this would result in error because in core Python there is no such function as sqrt. Okay, Does that make sense?
Installation is, sir, not enough, so we wanted to make it, matlab import is always necessary over there from
Yes,
our
yes.
side.
So installation is that NumPy is
Mm.
now there in your computer, but
Okay.
it's there on your computer, not in your program. So to bring it from your computer to your program, you need to run this command, import whatever library you want to import.
Hello, sir.
Yes, yes.
I just wanted to ask, going ahead, when we go for actual, when we do bioinformatics analysis using Python, so for every command, do we have to install specific libraries before we can start using individual modules? If it's
Not.
not in the core, if all the commands are not present in core Python,
Uh-huh.
then so we have to install specific libraries to do the bioinformatics in future.
Yes, yes. So if you want to use Biopython, you have to install Biopython. If you want to use some other library, you have to install that library. Yes. But installation is a one-time thing, of course, right?
Okay.
Yeah. So once you have installed Biopython, you don't have to install every time. Okay.
Sir, matlab you wanted to say that we have to, matlab after installation also we will not able to get, so we have to always import so for from your computer, to get into computer.
Yeah. See, installation, you install a program, and then you have to, just in other programs as well, this is very common. Like for example, if you have installed MS Office, okay. Now MS Office is installed in your computer, but when you want to run Word, you have to click Word. When you have to run PowerPoint, you have to click PowerPoint. Only then those programs will open up, right? Although they are there, they don't just open by themselves. So same concept here, that you can install as many libraries as you want, but when you want to use that library, then you need to import that library. Okay.
Matlab using part you are saying we have to get into again. Okay.
Yeah, so we have to tell the program that we want to use NumPy. That how else would program know that we want to use Biopython? There is no other way.
Okay.
There has to be a way to tell our program that we want to use Biopython. So that way of saying is import that library name. Okay. Okay. So now we go to the Biopython part. Uh, when we say import bio, now once you have installed Biopython, then we say import bio. So this bio is the library, Biopython library's name is it's imported by bio bio keyword. And then just to check whether installation is correct and the library is working, we say print bio dot and then two underscores version, then two underscores. Okay. So if you run this, you will see the version number. So I hope you are getting the version number. So if you get the version number, that means Biopython is installed and everything is working fine. Okay. Now, so first we'll see that, I mean, when you're working with bioinformatics data, most of the time is sequences, be it DNA sequences or protein sequences. So let's see how we can create the first thing we need to do is, as we talked about, that it's object oriented. So we need to create a sequence object, because once we have a sequence object, we'll have certain functionality available to us. So let's see how we can create a sequence object. So we say from bio.seq, S capital, B capital, S capital, import seq, S capital. Okay. So this is the function we are importing seq function from bio.seq. Let me just comment these. And then we say new sequence. This could be any variable name. Okay, this need not be new underscore sequence. You can just say X also, equals to seq this function, and then whatever sequence you want within quotes, and then you can do print new sequence. So it would print the sequence. Now you might think that, okay, this is similar to strings. So if you can similarly could have done s x is equal to this thing and then print x. This would have given the same output, but the difference is, if you do the print type new underscore sequence and print type X. So the sequence new underscore sequence is an object of bio.seq.seq. Okay, while X is an object of class string. Right. So that's the difference between these two. Now, what this difference really means, I mean, in terms of programming, what this difference means? This means that there are some additional set of functions available to us when we are working with sequence object. So let's look at some of those functions. Let me just clean this up. So first, we have this new underscore sequence object, and then we say dot complement. Now we know that DNA is double stranded. So if we have a sequence of one strand and we want to find the sequence of the complementary strand, we can just say new underscore sequence dot complement. And if you run this, this is this print new sequence, this one, and then the sequence of the complementary strand. So A becomes T, T becomes A, G, C, A, T, and so on. Okay. Another function that is available for sequence object is translate. So if you have in this case, like we have a DNA sequence and you want to computationally translate into a protein sequence based on the codon. So it would take first 3 nucleotides as one codon, find out its amino acid based on codon table, and next 3 nucleotides, and so on. So if you run this, we'll get this tetrapeptide, asparagine, tryptophan, asparagine, leucine. So that correspond to these triplets. Okay, so this was about basics of a sequence object. Now, when we are working with sequences, we don't manually type sequences like we have typed over here. We generally have sequences from some database, or we have some file that we want to read. So let's see how we can read a file. So first, you can, if you have a sequence file, you can use that. If you don't have, then you can go to this link and download a sequence file. This is a sequence file with multiple FASTA sequences. So you can just download this, and you need to save it. I mean, I have given this name kinases.txt. You can give any name, or you can give kinases.txt as well. And then where do you want to, now next point is, where do you save this file?
Excuse me, sir.
Yes. Is there a question?
Excuse me, sir.
Yes.
I just wanted to ask, what is the syntax used for? Yes, sir. I wanted to know what the syntax was used to find the complementary strand sequence.
Okay, so the syntax was first you create a sequence object for whatever sequence you want to find the complementary strand, and then you say print that sequence object dot complement. So if you run this much only, you will get the complementary strand sequence. This complement function is used to get the complementary strand. Okay.
Okay,
Alright.
sir. Thank you.
Yeah. So
Sorry to interrupt
no,
in
go
the
ahead.
middle of it, sir.
Yeah,
But
go
I've
ahead.
been trying to use the command import bio, but
Uh-huh.
constantly it's showing no module named bio.
So have you run
What
this
to do about
command
it?
pip install Biopython?
No, I haven't. I'll have to.
So maybe you should run this command. So just, I mean, open a blank cell and type this, and then run this. So this should install Biopython.
Excuse me, sir.
Yes.
Sir, in translate, when you did, then only that amino acid peptide chain came over there.
Yes. See.
Fine. That is the syntax for that peptide one.
Yeah. So if you want to computationally translate a nucleotide sequence, you can just use the translate function.
Okay, fine, sir.
Okay. All right. So now where do you
So you have imported differently for both the mod. I mean, I can see here that from bio.seq import seq, and from the
right?
next it's just from bio. Can you tell me the difference? Why
I will
is
go to
that?
that. We'll go to that. We'll go to that.
Oh, okay, sir.
Let's first save this file first. So where do you want to save? I mean, where you should save this file is that just type pwd present working directory. I mean, just pwd in a cell, blank cell, and run this, and there you will see a path, c colon something user something, or if you are on Linux, then /home/user something. So save this file in that path. Okay, otherwise you will see file not found error. So name it, give it some name and save it in the path. Okay. Next, okay, so now we want to read file. So in this case, we'll do from bio import seek.io. So now this, I mean, as the question was asked, that from bio.seek import seek, and from here we are doing bio import seek.io. So these libraries, so they have, I mean, when you talk about object-oriented programming, that basically means that there are classes. So seek is a class in bio library. Similarly, seek.io is a class in bio library. So depending upon what kind of functions we want to include or import, we can use these kind of constructs. So for example, in this case, we are saying from bio libraries seek class import seek function, and here we are saying from Biopython library import the seek.io class. So these are the differences when you import in this manner. You can either import the function, a particular function, or you can import an entire class, or you can import an entire library, depending upon how much you want to import. So so once we have imported this seek.io function, this seek.io means sequence input output. So through this function we can read and write files. So we say for all underscore seeks. So this is a variable name. Now, okay, this is a for loop part. So this for loop here means, I mean, when you are iterating, let me just quickly give you an example of for loop, how for loop works. We have not covered that. There's a chapter on loops and conditionals in the book. You can go through that. I in X print I. So when I do this, I have a list X equals 1 2 3 4 5, and when I want to iterate through this list, that means when I want to do something on each element of this list, I say for some variable name. In this case, I'm saying I in X print I. So here this will first assign a value of 1 to I, and then whatever code we have, we can also say, let's say I multiplied by 2.
Sir, how we will know where the for loop ends?
That's what. Okay, so when we say for I in X, so this X, whatever that list is, it would start from the beginning and go till the end of X. Okay, so this start and end is the X part, the list part. So we say, yeah, okay, so basically we are iterating through each element of this list. So unlike other C++ or other languages where we have start point, end point, so this is slightly different syntax where you just say one by one take element of X. In simple terms, you can say that.
No, I was asking that we have brackets in a different language, right, for loop.
Yeah.
Here till X, like it will.
See, that is a different syntax where you have initialization, increment, and condition. Three things are there in a for loop. This is slightly different. Here we are saying one by one take element from X. That's in simple terms you can understand like this.
Okay.
So you have a list X, and then we are saying one by one take element from starting from beginning, take element of X and assign it to I.
Okay.
Okay, so for I in X colon print I multiply by 2. So this is how you iterate through a list. Okay. So I hope you have saved. Yeah. Is there a question?
I have one question here.
Yes.
So
Seek
how do you save
Wait.
that?
Somebody was asking before you. Karmika?
Yeah.
Yeah.
So in this seek.io, we have given the file name kinase.txt. Right?
Yeah.
So, but user can give any name.
Yes.
So we have specifically mentioned here kinase.txt. So how we can make it dynamic, like it can pick any name from there?
Okay. That's a good question. Okay. Just wait for this question. Let me just complete this part because there are a lot many concepts in your questions that you have asked, so I have to cover those. Yeah, let me take another question. So there was another question somebody was asking.
Uh, hello.
Yeah.
Yes, sir. Sir, you said that saving part we are using PWD, but that is our present working directory. So how matlab, that is for saving part?
No, saving part is that if once you have downloaded this file, okay, now where do
Hmm.
you save it? On desktop, on C drive, where do you save it? So that part
So,
is the PWD part.
uh,
Okay.
so we have to provide file name also, sir, with that.
Yeah, see, once you go to this link, you get us some sequences, right? Or you can just right click and click save link as. But then you have to give some file name and also location. Where do you want to save? I mean, you can save it on desktop, save it on my documents, C drive, D drive. So where do you
Okay,
want to save it?
matlab that kind of matlab we have to give only the saving, not the PWD command we have to
No,
give.
no, not the PWD command. That is just for your information that which location you should save. That's the
Okay,
thing.
okay. Okay, actually, I thought that that was the present working directory, so I thought that thing.
No.
Okay, fine, sir.
Okay. Okay. So yeah, I have noted your question. I mean, I'll talk about it. There are a lot many concepts in dynamically assigning name, and there are multiple ways to do that. You can either take user input or you can uh, take some command line arguments. So we'll cover that. Okay, let me just finish this box and then I'll go to that. So from bio import seek IO, and then we say for all sequences, all seeks. This is a variable name. This could be anything. You can just say X in seekio.parse. So this parse function is used to parse the file, whatever sequence file you have. Now here we are saying kinesis.txt. This is the file name, comma. Then we also need to give the format in which we have sequences. In this file, the sequences are in FASTA format. So that's why we are saying FASTA. In case they are in some other format like cluster W format, then you have to say cluster. Okay, and then we say print all seeks dot description and then print all seeks dot seek. I mean, all seeks is this variable name, whatever this variable name is. Now, if you are familiar with FASTA format, at the top there is a header or annotation part of the FASTA, and at the bottom is the sequence. So whatever is the header part, that goes into description, and whatever is the sequence, that goes into the seek. Okay, so if I run this, I get all these. Whatever the file was, the entire file I'll get. Let's say I want to print only the description, only the headers. So I can just comment this part and I'll get all the headers of the sequence. Okay, so this is how we can read the files. Now, coming to this question on how we can change these files. Okay, so there are there is another library called sys library. So we say import sys. Now you can take command line argument. Okay, so for example, this is slightly advanced. Okay, so I'll answer your question, but so when you're running on a terminal, uh, so there you can, while you're running your program, you can just specify the file name as well along next to the program. So when you run, so import sys, and then here instead of file name, you can just say sys.argv1. So this is a command line argument, uh, and this you can specify when you're running on a console. This, I know this is slightly advanced, but maybe you can go through the book. I think in files chapter I have discussed this, so you can read through this. I mean, how it executes. So this is the way of asking the user the file rather than specifying within the code. And of course, most of the time we do not specify file within the code. We ask user that file name, because file name of course can change every time. Okay.
For loop is used over here or not in this program or not?
What?
For loop which you have done above, that is used in this part or not? See?
This for loop.
Huh.
No, this is not required. These are some just to give you an idea of for loop.
Okay, sir.
Okay. So just like we can read files, similarly we can write files as well. So for example, if you want to write, let's say, top 10 sequences. So this file has thousands of sequences, and let's say if you want to write top 10 sequences from this file to another file. So first, what we can do is we can create an empty list. So we calling it all underscore seeks. This again, this is a variable name. This could be anything, and then we say for the same for loop for seek underscore record in seekio.parse file name, comma, file format, colon, and then instead of printing the sequences, here we printed the description and sequences. Instead of printing, we say this list all underscore seeks dot append, and then we give this variable name, whatever we have given over here. So if we do this, let's see this much. So we say print len all seek. So we are printing the length of all seeks object, and length is 5489. So 5489 means there are 5489 sequences in our kinases.txt file. Now, if we want to write let's say 10 sequences, top 10 sequences, then we can say seekio, and instead of parse, we'll say write, and then this list name, and then a slice. Remember the slice operator. So we are basic 0, basically start from the beginning. We can skip the 0, colon, and go till 10. That this would be total 10 sequences, 0 to 9. So this is, so it won't save all the sequences. It would save a slice of this list, that is the first 10 sequences. So write function takes 3 arguments: the sequences that we want to write, the file in which we want to write. So we need to give a file name. So this file name would be created. Okay. So test, we are saying it test.aln, and then we need to specify the format in which we want to save. Here we are saying format is cluster format. So let's run this. So it has now saved the sequences. So if you want to check, we can just do load. So this command is works only in Jupyter. It's not a Python command. Test.aln. So if you are on any other IDE, this won't work. So this, look at this. So this has now saved first 10 sequences in cluster format. We had the file in FASTA format. We created a list, and then we took a slice of a list and save it in a cluster format. Okay. Now this was about reading and writing sequences, individual sequences. Now many times we are working with multiple sequence alignments as well. So let's see how we can read alignments. So syntax is pretty much similar. There is one difference that instead, when we are working with alignments, we'll use the align.io class instead of seek.io. So we say from bio import align.io, and then we'll create an align.io object. We call it align1 equals align.io.read, and we give a this file name. We can use the same file because this is actually an alignment file. So kinases.txt, and again we need to specify the format, alignment format. And if we do print align1, we get this.
I have one question.
Yes.
When we'll be using this align IO, which algorithm like cluster W or muscle it will be taking?
Okay, good. So align.io would not do any alignment. Align.io is only for reading and writing alignment files. So if you want to do alignment, you need to do some other program that is for alignment, be it cluster, tea, coffee, muscle, any program. You first align them, and then once you have then output from that alignment program, then you can use it in align.io. Okay.
Sir, can you
It
please
is only going to tell us about the length of the sequences.
No,
Or
it's
anything else?
a lot many things. We'll go to why we are using align.io. This part we'll cover in this one. Yeah.
Sir, can you please go to the previous command that you did?
Which previous one? This one?
Yes, sir. 37. Yes.
37, yeah.
So here you made a FASTA file into a cluster format. Something. I didn't understand this one.
Oh, you can follow? Okay. So the point was here in this that we want to read a file, sequence file, and we want to take a chunk of those sequences. That's it. Because in our sequence file there were 5,000 sequences, more than 5,000
Okay.
sequences.
Understood. Okay.
Okay, good. Okay.
And in the next one, then
Next alignment one. So now we are working with alignments, and we'll see how we can take a chunk of an alignment in this. Let me just explain. So
So, sir,
so what
align
is the
IO is
output coming?
I have not yet for the alignment part.
No, this output that has come. What is this output?
This output.
Yes.
Yeah, I'll explain. Wait, this output I'll explain.
Okay.
Yeah.
excuse me, sir.
Yes.
Sir, align IO is using which algorithm?
See, I just answered that question. So align IO is not for doing alignment. Okay, this is very important. Align
Okay.
IO is only to process alignment files. So if
Okay,
you
I
want
missed.
to, sorry,
Sorry, sir.
yeah, go ahead.
Sir, that time I missed that point. That's why,
Okay,
sorry.
that's okay, no problem. But align IO is not for alignment, it's for processing alignment results. So what kind of processing we'll see now, that what kind of
Okay,
processing we can do. Okay.
okay,
So
thank you so much.
sure, so far we have done is created an object align1 and read an alignment file, and we print this alignment file. Now look at this output. So this is the output. So here each line is one sequence. So this is entire one sequence starting from the end. In between there is dot dot dot. It's quite a big sequence, but it has truncated this and written triple dots. And this is the sequence header, whatever the annotation is. So now this alignment file has, so it says alignment with 5489 rows. So it has 5489 rows, which means 5489 sequences and 1094 columns. So the alignment length is about 1000, and the number of sequences is, we have is around 5500. So this alignment is now like a matrix. Okay, you have rows and you have columns, where rows represent sequences and columns represent the alignment positions. Now let's say we did this alignment to study some motif, for example sequence motif. Let's say we want there is this, let's say these are kinase sequences. So let's say there is some ATP binding motif that we want to study, and we want to study the highlight the conservation of ATP binding motif, that across these 5000 sequences, how does our ATP binding motif look like? Okay, that's our question. Now how we can answer this question, or how we can process this alignment to answer that question? Okay, I hope you have understood the question part. Now
Sorry, sir. Can you
yeah,
pardon again?
sure, go ahead. Is there a question?
No, sir. Would you pardon? Actually, I haven't understood this part
Okay,
properly.
the question part. Okay, what I'm saying is that so far we have an alignment file of this file, and we have created an alignment object using align.io.read, and that's it. We have printed that object. Now the question is that using this alignment object, we want to answer some questions, because that whenever you do alignment, multiple sequence alignment, you have some question in your mind that, okay, I want to answer this question, that's why I'm doing alignment. Okay, so in this case, let's say the question is, we want to study the sequence conservation of ATP binding motif. Okay, I hope this part is clear. Now let's say that ATP binding motif is from here to here, just for example. Okay, so in all the sequences, this column, these columns, this entire thing is ATP binding motif. Okay, now what we want to do is, we want to extract this chunk, this slice from the alignment, and save it separately. We don't want the entire alignment, because if you have this big alignment with 5,000 rows, 5,000 sequences, and these many alignment positions, it's almost impossible to have this entire alignment in your report or a dissertation. But if you have some chunk of this alignment that you want to show, that you can do easily. So let's see how you can extract the slice. Okay, I hope the question is clear. Now, so first we'll extract a slice of this alignment. Let's see how we can do this. So this previously the slice that we have seen was a 1D, one-dimensional slice. We had a string that's one-dimensional and we took its slice by giving start position and end position. Same concept we'll use here in alignment object. The difference would be now we have a 2D, two-dimensional string instead of a one-dimensional string. So for that, the syntax would be we'll first create some variable name x equal to this alignment object that we have created, and in the slice we'll say first the row range, that means the start and end position for the rows, and then comma start and end position for the columns. Okay, so in the row range we are saying :10, that means start from the beginning, go till the 9th sequence. So it would start at this row and 1, 2, 3, 4, 5, 6, 7, 8, 9. This it would go till here. Okay, that's our row range. Column range we are saying 3 to 3:12. So this is 0, 1, 2, 3. So our slice for the column would start at this L column, and 3, 4, 5, 6, 7, 8, 9, 10, 11 till A. From this L to A part, from all the sequences, and for the first 10 sequences would be taken. Now let's print X. I'll comment this one.
So I have one question.
Yes.
What if I don't know the position? Let's suppose
Correct.
I have 1,000 base pair and I want to look for RXLR motif, right?
Hmm.
So there will be two variables. X I don't know, R I have to look for, and LR, right? So how I can do that?
Sorry, RXLR. What is RXLR?
This is pathogen.
Okay, so you have a motif called.
RX
Okay,
X can be any amino acid.
I see. I see. Okay, so you are saying you have some motif RXR.
Any motif, let's say any
Yeah,
motif.
yeah. Okay, any
We don't know
motif.
the exact position, but we have 1,000 base pair sequence, so we don't
Correct.
have to look manually. Rather, the software or the program will find it for me.
Okay, so I mean, program can find, but since you have a motif with you, you know the motif. One thing you can do is you can just open your alignment in a text editor, and for let's say in some at least one sequence, if you know the motif, just note down the position in that sequence. Okay, because the same position, if in one sequence you know that okay, at from residue number 509 to 515 that motif is, so you just note down in one sequence and you can specify in the slice, because you need to have some information about what chunk you want to take, and
Okay,
that.
so it's just for slicing and not for searching.
Yes, correct, absolutely correct.
Okay.
Yeah. Okay, so when we do print X, so it prints this slice. So look at now this sequence. Now it says alignment with 10 rows and 9 columns. So these are the first 10 sequences, and we have taken this 3 to 3:12 chunk from our alignment. So now this entire thing is very much presentable in a in a in a report or a dissertation or a manuscript, whatever you're working on. So this is and you can also of course write this part into a file in a, let's say, in a cluster format, and if you load this, so you'll get this. So you see, so you started with these 5,000 sequences in FASTA format alignment, and you have now created a 10 sequences in cluster format. So these kind of processing you can do with programmatically very easily. Manually this would have been a very painful job. Okay.
Hello, sir.
Yes.
This is quite applicable with what you showed. But using one more thing, I just wanted to ask for the multiple sequence alignment.
Uh-huh.
This is yeah, we can definitely use this. But if we want to like you have in the first row, probably you have the consensus sequence with reference to which the other rows are being compared.
Correct. No.
And so
Yeah. Yeah. Go ahead.
yeah,
Go ahead.
so so if I have such a consensus sequence and I want to know the match percentage of the subsequent rows, can we also do that?
Match percentage. I think
Means
that is, yeah, that can be done through a alignment program, not in Python. But
okay,
whatever program you're using, they generally give these pairwise similarity and all those things. Those statistics, alignment statistics, comes when you are doing the alignment part. So here we are not doing the alignment; we are just processing the results.
so that is not in this module.
No,
This
no. Align IO, there is no because Python multiple sequence. You can, for example, if you want to use cluster, so you can run cluster from Python, but Python there is no inbuilt program. You can run tcoffee from Python. That option is there, but the algorithm or the program would be that different program itself.
okay.
Yeah.
Sir,
Yes.
sir, can I also check mismatches between the two files?
Mismatches between two files.
Like one is the FASTA file, the other one is kinases.txt file.
Uh-huh.
If suppose there is a base pair change, so can I find it using this?
No, see, kinases.txt is a FASTA file.
Okay,
This is not, they are not two different files.
okay,
This is the file name and this is the file format.
format. Okay.
Yeah.
So can I compare two FASTA files?
You can compare, I mean, what sense you want to compare?
No, so suppose I have variation in the genome and I want to compare it with the reference. So what is the change of base pair in my test sample compared to a reference?
That's more like an alignment thing. I think you want to do pairwise alignment. That's because if you have two sequences, you want to see where they are different. That's the
Yes,
question, I think. Yeah, so that's
yes.
alignment. That's just alignment. If you do alignment of the two sequences, you will get to know where they're different.
Okay.
Doesn't?
So okay. So will it come in the output that there's a mismatch at this position?
Yeah, if you have any two sequences, you align them, it would show you where there is a mismatch and where there is a match. The alignment output is, that's what alignment is all about. Yeah.
Okay, sure.
Yeah.
Thank you.
Sure.
Sir, this is for splicing only, no? Nothing more than this, I think
No,
so.
no, just slicing. Yeah, we are just extracting the chunk of the alignment that we want. Okay. All right. All right. So moving on. So let's say you want to run blast search using Biopython. So when you want to run blast, you can either go to ncbi website and run blast program there, or you can download blast executables also. But if you download blast executables, you need to download the databases as well, and you need to also worry about updating those databases. Now in Biopython there is an option that you can run blast search through your program, and the search would be done on ncbi website. Okay, so let's see how we can run this thing. So to run blast programmatically, we'll need this from bio.blast class. We import these two functions, ncbi www and ncbi xml. And then to run blast, we'll first create a variable called result underscore ncbi. This could be any variable name. And then we say equals to, this variable equals to ncbi www dot qblast. Now qblast is a function that actually does the blast search, and it requires at least three arguments. First is the blast flavor that you want to use or blast program that you want to use. In this case, we are doing blast n, which is nucleotide blast. So it would search a nucleotide sequence against a nucleotide database. So if you are searching for a protein sequence against a protein database, you will use blast p. Now, the second argument is the database against which you want to do the search. Here I have specified NT. So NT is nucleotide database. So here you can specify any database that is available on NCBI website. And then third argument is the sequence that you want to search. So either you can give in this, like I have given GIID. So this would take the sequence from the NCBI gene database, or you can specify sequence file also. If you scroll down, you will see that example for how you can input sequence file. So this command, this statement, what this would do is it would take the sequence corresponding to this GIID, run a blast and search against the NT database available at NCBI. So this would run this command, and then once we have executed this command, the first thing you should do is you should save the results in a file. So for that, we'll say with open. Again, this is file handling. You can read the file handling chapter for more details on this. So with open, then here we are giving a file name in which you want to save the results. And whenever we want to write something to a file, we give this argument w. And as file handle, so this basically is a syntax of writing any file. If you want to write any, instead of, let's say instead of printing, you want to write to a file, this is pretty much the syntax. So then file handle dot write this variable name dot read. So this would now, after this you have executed this, it would create this file. So let's run this. This would take some time because this would take your sequence and run it on NCBI website. So this, and when it runs, you will see an asterisk over here. So let it run. And once this is complete, this asterisk would change to a number, and we'll have this file, my_blast.xml, in our PWD, present working directory.
Sorry, another question. So
Yes.
if we want to compare two sequences, so instead of TA, we put the another GIID over there. Will it pick BLAST2P?
BLAST2P, I think we have.
BLAST2N, not P, sorry, if it's nucleotide then.
Sorry, if you have two sequences.
See, here what it will do is it will take this GID and it will compare with the nucleotide database, right?
Correct.
And it will perform BLAST-N.
Yes.
Now I want to specifically mention I have two genomes. I want to compare those. All right. So
Okay.
instead of doing that, there is one another option in BLAST. If you have seen, you can specify two sequences so that you can do it from BLAST2P or BLAST2N. So
Yeah.
if I replace this NT database with another GID, so will it pick that BLAST2N or it'll, I mean, how will it do that comparison?
Okay, so first we need to check whether the blast, you're saying BLAST2N, that part is supported or not. I'm not sure if that is supported, because if QBLAST support this BLAST2N, then maybe you can specify two GIIDs. But if this BLAST2N is not supported, and that I'm not sure about that.
Okay.
So, is there another question?
No, thank you.
Okay, sure. So you can, I mean, you need to look into this manual, QBLAST manual, and see if it supports BLAST2N. Yeah. So it's taking longer than usual. I don't know. Okay, done. So this is done. So if you don't have any question, please mute yourself. Okay. Now, so this is done. So now we have this file my_blast.xml. So we'll reopen this file and save it in a variable name object, and then we'll parse this file to see the alignment, to see the results. Now blast object blast search saves result in a hierarchical manner. So we'll have a blast object at the top, then object would have alignments, and alignments would have high scoring pairs. Now this is like a nested list. So let me just quickly show you what I mean by nested list. So let's say we have 1, 2, and then 3, 4. So if you have this kind of a thing and we say print X, it prints the, so this nested list means list of lists. Now if I do for Y in X, print Y, again it would take this is the first element 1 comma 2, 3 comma 4 is the second element, so it prints those elements. Now let's say I want to print 1 2 3 4. So in that case I'll have to run 2 for loops for, say, in Y print, so that's how I can get 1 2 3 4. So when we have nested list, so then we have to run multiple for loops. That's the key message over here. So in this case, since blast object is such a this kind of a object where you have alignments, and within alignments you have high scoring pairs, so we'll open this XML file and then have a variable name blast records and ncbi xml.read this file. Now we can say for alignment in blast record dot alignments. So blast record, this object has an attribute called alignment. And then, so this is another for loop. So as I was mentioning, nested list. So first list is blast record dot alignments, and within alignment you will have HSPS. So high scoring pairs in alignment dot HSPS. So if we run this, we can print alignment dot title for to see the blast output. So we get this. So we have the name of the hit and then some statistics, e-value score, and then the match mismatch that you see in the blast. And this is the second hit.
Excuse me, sir.
So yes.
Earlier program do not have shown the output.
No, because it has just saved the file.
Okay, okay, read file. Okay, read
Yeah.
command.
Yeah. So we are saying just we are doing this right now. Write. So write would just write the file. There is no output here. Okay.
Okay, fine.
Okay. So
Sir,
now
previously, is it the accession number that we need to provide while doing blast?
either accession number or sequence file.
Okay.
Yeah. Now the whole point, why do you want to do it through Python? Why? I mean, you can simply go to NCBI and do it over there. The thing is, this, I mean, when you're working with this kind of a programmatic access to the NCBI servers, you can design your protocols and you can have your customized results ready. For example, in this output, let's say you want to apply a e-value cutoff and you want to see only those results which satisfy that e-value cutoff. So we'll have, for example, in this case, we are writing to a file. Let me just comment this part and just straight away print over here, or we can go with this, that's okay. So we are saying with open new file, some file name in write mode as file handle. So basically this is just to write a file. Even if you don't have this, that's okay. So for alignment in blast record dot alignments, the same for loop that we saw previously, and then for HSP in alignment dot HSPS. Now instead of printing the results, we'll add an if condition. So you can read about loops and conditionals where I explained how this if condition works. So if I say this HSP, this object dot expect, so this HSP has an expect attribute that has the e-value. So if I say if this e-value is less than 10 to the power minus 105, then print the HSP and also write to a file. Again, this is writing to a file. So if we run this, we'll have this output. So see, previously we had lot many hits. Now from that output, we have now only 3 hits. And if you look at the e-value for all these 3 hits, it's lower than 10 to the power minus 105. So depending upon what you're doing or what kind of protocols you want to set up, so this way, let's say if you are working on some pathogenic drug targets, let's for the example, and you want to find out human homologs. Let's say you have 10 targets and you want to find out human homologs. So you can create. Could you please mute yourself? Yeah. Yeah. So point is that. So could you please mute yourself? Who is this participant? I don't know. Can I? Okay, I think, yeah, okay, I can mute. Yeah. Okay. So the point is, you can set up your protocol. That was the thing. So if you have, let's say, 10 drug targets, and for those targets from a pathogenic organism, and for each of them you want to do a blast search and find out human homologs. That's what you want to do. So one thing would be to going to NCBI site, doing search and filtering results. Here you can automatically set up a program for loop, and one by one it would do blast search and save all the results and give you only the human homologs. Now you can try this. This is an example where I have this protein sequence. Again, previous example was on nucleotide sequence and using GIID. Now let's say if you have a sequence in a file. So save this sequence in a file. For example, I have this sequence and save it, let's say example1.fasta. Now you can, when you are taking sequence from a file, there is a slightly different syntax. You say some variable name, open that FASTA file, and the qBlast, since this is a protein sequence, we'll say blast P, and our database would also be some protein database. In this case, I'm using non-redundant database, NR, and then instead of GIID, we can give this file as the sequence file. Now this sequence file can have more than one sequences as well. So you will, in a batch mode, you will be able to run blast search. So seq file.read, rest of the things remains the same. So here, I mean, in this example, the exercise was that you have this sequence and you want to find out from which organism or what is the most similar sequence for this protein sequence. I have removed the FASTA header here because so that you don't know what organism this one is from, and that's an exercise. So to figure out its organism name, we can do blast search, and once we have done blast search, we can just print the top hit. So the idea is, when you have this sequence and you search against a non-redundant database, the top hit, if this sequence is there in the database, the top hit should have 100% match, and that's how we can get to know that from which organism this is. If we get a 100% match, that means this sequence is from that organism. And if you run this part, you will see the top hit. I mean, top hit is, we are using this, instead of doing for loops, we are just saying square bracket 0, because the 0th element of the list, which is the top hit, because results are sorted by score. So if you run this, you will see the output, and in the output you will see the organism name, and that would be C. elegans. Okay, so yeah, I mean, there are some other things. For example, if you want to filter results, we saw filtering based on E value previously. If you want to filter from all the results, let's say you want mouse homologs, you have done some blast search, and from that search you only want mouse homologs. So you can apply this kind of a condition, or you can change whatever condition you want. You can have multiple conditions also. You can have E value or multiple organisms, so that those conditions you can set depending on what you are doing. And finally, there is a library, CSV library, to save these. I mean, in a CSV format, blast results in a CSV format. For example, if you are making some report or a dissertation, there you want to show blast results. So instead of taking screenshot of NCBI website, it's better you create a table that would have title, alignment title, organism name, similarity, e-value, score. So that table would look nice, much better than the screenshot. So that's Yeah.
Excuse me, sir.
Yes.
Sir, it will give the blast output in XML file format.
Yes.
Earlier, like.
Yes.
Okay.
Yeah. So, but you don't really need to manually read the XML file. You can just programmatically read the XML file. So it doesn't matter that format, what format it is.
Okay. Okay.
Because you will be just programmatically running it through for loops or conditions. So if it's an XML format or text format, it doesn't matter. Okay. Any questions, anyone on this? Okay. So if not, then practice all this, go through the other chapters in the book, and if you have any questions at any point, if you have any questions, feel free to
Excuse me,
email.
sir.
Yes, yes.
Sorry to
No, no, go ahead, go ahead.
interrupt you.
Go ahead, go
Uh,
ahead.
so there were one or two parts that I couldn't follow in this particular session. Could
Uh-huh.
we have the recorded session for this one?
Absolutely, absolutely. You'll have this recording available to you.
Okay, thank you, sir.
Yeah, yeah. All right. Yeah. So,
Sir,
yes,
sir, HSP is what? The full form of HSP over here, what is indicate?
high scoring pairs.
Okay. That is the value we are providing.
Yeah. No, high scoring pairs. I mean, this is a blast object that you have blast object. Then you have within blast object you have alignments, and within alignments you have high scoring pairs. So this is how
Okay,
it is. The blast results are stored internally by blast.
okay. That is e value or something also we can see.
Yeah, even e value and positives, all those things. The blast
That is
statistics that are there, they're all there.
blast force wise we can say.
Sorry.
Blast score, whatever blast
Yes,
score.
yes. Blast score, e value, whatever statistics comes with the blast, they are available to you here. Yeah. So yeah, and at any point, if you have any questions, practice these things. You feel free to email me if you have any questions.